up on your screen. All right, so welcome. Thanks for coming back. This is lecture number 2. We're going to talk about spawning and hatchery culture. This is actually a good one to do via PowerPoint because I can use pictures. It's hard to do, you know, it's nice to see the hatchery and everything in person, but it's hard to actually show all the components of this in person without doing an actual spawn. So I have some pretty cool videos that are actually separate from the presentation because I've never really seen videos embedded in the presentation pull off very easily. So once we get a few slides in, I will pull the videos up separately. So we're going to talk about spawning and hatchery culture. And I already forewarned you, there's a pop quizzy. So there's the pop quizzy. For those that were here last time or who watched that first video, the question is, after blank, an action or an activity for a blank period of time, we can then spawn the animals. Does anybody know the answer to any of those? Anybody fill in any of those blanks? Got a few veterans here. Come on, step up. Chris? Nobody, huh? Okay. So last time we talked about brood stock conditioning. That's the answer to the first blank. So after brood stock conditioning for 6 to 8 weeks, we can then spawn the animals. So I'm going to go back. So brood stock conditioning, for those that were here last time, is the technique we use to get the adult shellfish actually ready to spawn. So we can really do that any time of the year, which is pretty cool. So we start in the middle of the winter, which allows us to get a jump start on the season. So we start brood stock conditioning, which basically entails bringing the shellfish into the building, putting them in a spring-like temperature tank with lots of food, and the temperature and the food triggers them to develop their gametes, their sperm and egg, and get ready to spawn. So after that 6 to 8 week period, which we went over last time, they're ready to spawn. So let's do a quick review of the life cycle. So you have on this slide here on the bottom are all the adult shellfish, in this case oysters, but for oysters, clams, and scallops, it's all very similar. You have your adult shellfish on the bottom in this case, or in a spawning tank, which you'll see pictures of. They spawn, so sperm and egg are emitted into the water column. Sperm fertilizes the egg. Fertilization occurs pretty quickly, like within the hour. And then we can actually see under the microscope, and I have pictures of the egg dividing, which is really cool. 4 to 6 hours post-fertilization, we have this kind of a blob really called a trochophore, which is the really early larvae. 24 hours, usually in a hatchery where temperature and everything is ideal, it's 24 hours. We have what's called a veliger, which is a swimming larvae. And that name is derived from this organ here called the velum, which is what they use to swim around and grab the food particles out of the water and to feed themselves. So for the first 2 weeks or so, they are these veligers or planktonic swimming larvae. They're in the water column. And then after about 2 weeks, they do what's called setting. So they'll reach a certain size. They actually develop a foot, which is shown in this picture here, kind of an anthropomorphized shellfish standing up on its foot. Really, in truth, it'll be kind of laying on the bottom and using that foot to drag itself around. So in the case, and this is where all 3 species really start to differ. Before this, they're really all similar. They're sperm and egg, fertilized in the water column, planktonic larvae for a week and a half, 2 weeks, and then they set to the bottom. So in the case of oysters, they develop this foot, which is in this picture here. This could be either a clam or an oyster, really, with this big organ, the foot sticking out. They use that foot to drag themselves along the bottom and find a suitable substrate to set on. So in the case of oysters, they'll set, and they actually cement themselves to a surface, and then they lose that foot. So after they lose that foot, cement themselves, they no longer have the ability to move. Whereas clams, they set to the bottom, they retain their foot, and they keep their foot really forever. And for those of you that have eaten raw clam, that large, the big organ, the hard muscly organ in the clam when you eat it is the foot. And the scallops also retain their foot. It's much smaller, but they use that to attach to eelgrass blades and attach to the side of our tanks. So at this stage, once they set to the bottom, that's when we start to call them spat. You'll see the term here, early spat. So spat is the term we use for, post-larval, but before sexually mature shellfish, spat. So once the oysters set, before they set, they look just like this. They all look very similar, just like a miniature clam. And then when they set, they cement themselves and they spread themselves out on the surface and start to look like miniature clams or miniature oysters. So that's the life cycle in a nutshell. And y'all can feel free to shout out any questions you have. You can also raise, I see there's a chat. Oh, okay, good question. So what is the size of the larvae now? This is a very good question. So the larvae, we are 4 spawns in. So we've actually done 3 spawns of oysters, and we did our first clam spawn last week. So the largest oysters we have are already up to 3/16, quarter of an inch. They actually just went over to the nursery today. And the smallest stuff, the clam larvae that we have, are only about 1/10 or 2/10 of a millimeter in size.
Wow.
So when they set though, this is a good question. So when they set, when they start, the eggs are about 25 microns, a micron being 1/1000th of a millimeter. So, microscopic, thousandths of millimeters. And then when they set, they're about 1/4 millimeter. And then from there, they just take off. Good question. All right.
Thank you.
No problem. Any questions on that before I proceed to the next slide? Okay, so the spawning process, after that conditioning process that I mentioned, spawning involves this. We bring the conditioned, ready to spawn shellfish into the hatchery. So in this case, we have a spawning tank here, black for a reason. Anyone want to guess or speculate or tell me why the tank is black?
So we can see the eggs.
Very good. The eggs and the sperm are all white in color. So if this were a white tank, we really wouldn't be able to see anything. And you'll see, I have pictures of all this. So we lay them out in this nice grid. And the reason for that is we want to, you remember, these guys are protandric hermaphrodites, which means they're one gender at a time, but they can switch back and forth. So we don't know, in this tank, we don't know who's who or what's what until they spawn. But if you condition them long enough, you always have females. So we spread them out so we can keep an eye on things. We place them in this tank first thing in the morning at, say, 60 degrees. All right, springtime, and then we slowly raise that water temperature up to summertime temps. So we're making them think that it's summertime, so it's time to spawn. So that temperature increase, along with the addition of food, triggers them to spawn. And another old trick that we use is we can take frozen sperm from a previous spawn, put that in the tank, and that chemical inducement will get them going. So
This is my wife. I just want to interrupt because she wants to be included in our new oyster venture. This is Lynn.
All right, thanks for coming.
Hi, nice to see you.
Sorry to interrupt.
No problem. So say 9 o'clock, everything gets started. We get the oysters in the tank here, start to warm them up. And then really within a couple of hours, they'll start spawning. So when they start spawning, we will actually note who's a male and who's a female. we'll let the males spawn in the tank for just a few seconds because we don't want there to be too much sperm in the tank, because if you have too much sperm fertilizing the eggs, you end up with mutations. So we have to maintain a nice ratio of sperm to egg. So if we get males spawning, we'll take them out, leave the females in. So all the eggs will be in the tank and we can monitor them for development.
How can you tell the difference once they start to spawn?
Great
Which ones are
question.
male and which ones are female?
Great question. So the sperm is super small. It's absolutely microscopic. You not see the individuals with a naked eye. Eggs, on the other hand, are much larger. They're about 25 microns in size, which is tiny, but you can actually see the grains of the eggs in the water column. So we'll take a glass tube, a pipette, and suck up a sample, and the sperm just looks like milky water, basically, and the eggs, you can actually see the graininess of the eggs in the water column. And if my videos work, you'll actually see the graininess of the eggs in my videos. So I'm going to actually pull up some videos. This was supposed to be a video. I'll try and play it, but so this is a male spawning. You can see that kind of smoky plume coming out the shell here.
Evie, come.
I'm going to pull up a different video.
Evie. Steve.
I have to move this over to the other screen. Not gonna work. Not letting me do that. Hold on. Technical difficulties. So what I'm gonna do is stop sharing this, and then I'm gonna share, hopefully share my other screen so I can show you. All right, is that working? So there's a,
Yes, this is great.
okay.
We see it.
For some reason, like I said, when you embed these videos into PowerPoints, they never seem to work. So this one's staying overhead the way it's supposed to. So this is a male oyster spawning. You can see the sperm coming out and that smoky plume there. And that's very typical of males. And of course, you know, 9 times out of 10, every spawn, the males usually go first. Surprise, surprise, right? Folks in the room. The next video.
Scallops.
Next video is going to be a female. Let's see, where is that? I know this is one. So can you guys see the graininess in the water?
Yes.
So that's all eggs, and then this female just puffed a whole bunch of eggs out.
Mhm.
I'm gonna play that again. So you can actually see in the water column all that graininess, right? That's all eggs, millions and millions of eggs. Watch the puff.
Yep.
So the female, this is typical of females, they will actually puff the eggs out in these big clouds. I think I have a better one here. Yeah, this is a better one. Watch. Yeah,
Whoa.
right?
Wow.
Cool, huh? So yeah, this is typical of the oysters. The sperm comes out in that smoky plume. Eggs come out in these beautiful puffs.
So Barley, does this mean that the oyster to the right is a male?
Let me pull that video up again. No, the oyster to the right is either not done anything yet, or it's also a female. Because like I said, we wouldn't leave the males, we don't leave the males in the tank for long.
Oh,
Once we see them spawning, we let them spawn for literally 15 seconds or so.
all those little white dots are the male sperm, correct?
No, those are the eggs.
I mean, okay.
The sperm we cannot see. The sperm is like smoke, smoky water.
Barley, can you tell the difference between male and female just by looking at the shell?
No, you cannot. That's the trick. So when we condition the shellfish, we condition about 30 of them at a time. And we always get a nice ratio of males to females. So fecundity, which is the biological word for number of eggs or, spawnability of the animals, the fecundity of these things is phenomenal. So oysters and clams, when they spawn, these females are producing millions, tens of millions of eggs. So all we need are a few females to go at each spawn, and we'll have plenty of eggs. So the reason they do that, just like most things out in the wild, they produce millions and millions of offspring in hopes that just a few percent will survive. So in our case, we get much better survival because everything's contained. I'm going to show you a female clam spawning. So that's, like I said, it's a female, so that's all eggs coming out. And the fecundity that I just mentioned, you see this huge pile of eggs on the tank, right?
Mm-hmm.
Literally tens of millions of eggs. This is a pretty large female here. And this one, you notice we have an F on this one. That's because we spawned it previously. So we knew it was a female. So we marked it and then reconditioned it or got it ready to spawn once again. I'll play that again.
Just another question that you've surely already told us the answer to. What is the stimulus to make them do this? Both the male and the female? Was it the water temperature that
It's
triggers it?
water temp after they're if they're ready to spawn, if our conditioning works the way it should, which it usually does, if they're ready to spawn, the spike in water temperature and the introduction of food, as well as that other trick that I mentioned, which is the introduction of frozen sperm, gets them to spawn. So in the wild, they're starting to condition right about now. If you remember, the magic number for shellfish feeding is about 50 degrees. So any temperature below 50, they pretty much shut down. They don't really feed. Temperatures above 50, they start to feed. So between 50 degrees and 65, 70 degrees is their conditioning temperature. That's the spring life temperature when they're just coming out of dormancy. They're going to start to feed, and they're putting a lot of their energy toward getting ready to spawn. And then once we get that first pulse of hot weather in the late spring, early summer, usually early June, then they'll spawn naturally out in the wild. So we're getting a jump start on the season by manipulating things here in the hatchery.
When they
Farley,
spawn in the
so-
wild, by what degree is it more productive? You have more fertilization here in these controlled circumstances, and in the wild we have massive dilution and all sorts of other things. Is it like a 10,000 times more effective than the wild?
That's a good question, and yeah, your points are right on. You have if the adults aren't close enough together, which is one of the reasons we think that shellfish aren't making a robust comeback. If the adults aren't close enough together, if you have a female on one end of the kitchen table and a male on the other end of the kitchen table, and they both spawn, their sperm and eggs might not meet. Whereas if they're close together, there's much better chance of fertilization. And then you have all the predators, because like I said, they're planktonic larvae for the first 2 weeks or so of their life. And there's tons of stuff out there that's either filter feeding, like bunker, for instance, that are or herbivores that are constantly swimming through the water column with their mouths open. And then if they do make it through that planktonic stage and they set to the bottom, there's all the stuff on the bottom that's going to try and eat them. So like I said, if you get 1 or 2% survival in the wild, you're doing pretty well.
Farley, are the clams either male or female? I think you said the oysters can switch off from one to the other, but then you said you marked the female clam. Are they different?
Clams and oysters are both the same. They're what's called protandric hermaphrodites. So that means that when they sexually mature, they're generally males, and then they switch to female. But if needed, they can switch back and forth. So if you were to take a population of Now, if you were to take 20 males, for instance, and put them in a bag and put them off somewhere on their own, they would then differentiate so that they were a mix of males and females, so that they, when they spawned, they would actually produce offspring. Scallops, on the other hand, which let me start showing some videos of those. Scallops are simultaneous hermaphrodites. This is just a fun picture of a scallop swimming in the spawning tank. And this is a picture of a scallop spawning. Again, you can see the puff of eggs in the water column. You see that one?
Yep. Mm-hmm.
So scallops are simultaneous hermaphrodites. So they produce sperm and egg at the same time. So, and during spawning, they'll switch back and forth. So scallop spawning is pretty hectic because this scallop here might start spawning sperm, and then a few minutes later, it'll switch to eggs. And because we don't want too much sperm in the tank, we're constantly taking scallops in and out of the tank and trying to keep track of who's who and who's doing what. But they don't self-fertilize. So there's got to be a nice mixed population. So scallop spawns are pretty exciting because they do all this moving around. Let me show the swimmer again. So
With
for
the
those
eyes.
that didn't realize or forgot, scallops can swim, obviously.
Mm-hmm.
They have eyes.
They have eyes.
You see these little blue dots on the edge of the shell.
Mm-hmm.
Can you explain again, can the sperm double fertilize the eggs or something if there's too many in there? What was that about? Why you need to take the males out?
Yeah, basically what happens is if we have too much sperm in the spawning tank, I guess I can go back to the PowerPoint now. If we have too much sperm in the spawning tank and too many sperm fertilize the eggs, then they don't develop properly. They end up mutated. We call them abbeys, as in abnormal. Remember old Frankenstein?
Mm-hmm.
Abby normal?
Abnormal. Abnormal from
Right, I like that,
Frankenstein
Abby
movie.
normal.
We call them abbeys.
Good name for a cartoon character.
Abnormal. So these are basically just the beginnings of the videos. See all those eggs. So this is the bottom of an oyster spawn tank, pretty much at the end of a spawn, covered in eggs.
We don't see anymore.
Excuse me,
We lost the
Marley,
video.
we don't
Oh,
have a
whoops.
video, yeah,
I gotta
we just have
reshare.
your town of East Hampton.
Okay, I gotta reshare. Gotcha. Stop share. Okay, got it?
Yeah.
Yes.
Yes.
Okay, so this is a picture at the end of an oyster spawn with the tank, the nice black tank full of these beautiful white oyster eggs. And like I said, if we get a good spawn, we'll end up with literally hundreds of millions of larvae the next day, which is way more than we need. But that's all right because the females produced a lot of eggs. They were happy. So what happens? So now we'll get into the nitty gritty of what the stuff actually looks like up close. On the left, you have eggs right after a spawn. So these are shellfish eggs. They're about 25 to 30 microns in diameter, which is 25 hundredths of a millimeter. And these are nice, round, properly shaped eggs. And I don't know if you can see on your screen, but there's kind of a yolk sac around the egg. And then on the right-hand side, really within hours, we can see the initial division. Does anybody who took, any biologists in the audience or anyone who took college biology, you might remember the term polar body. The polar body is the initial division point on the egg. It's the point where the egg starts to divide or to cleave. So the first thing we'll see is a polar body, which is basically a little speck on the edge of the egg, and then they start to divide, and they look like these eggs on the right-hand side. So this is about 2 hours after the spawn. We have beautiful cell division. The day after, we have these de-hinged veliger larvae that I mentioned before in the life cycle picture. So in this picture on the left is a beautiful de-hinged, could be clam, scallop, or oyster. They all look the same at this point. And this organ here on the right is the velum, and that's what they're using to swim and grab the food. So the day after so once the spawn occurs and once we see that the eggs are divided, we put all that material into one or two of these 100-gallon conical tanks. And then the next day we siphon those eggs out of the tank. So you can see on the right-hand side, this is a piece of pipe with a very, very fine mesh on it, where we're siphoning those eggs out. And these, I can tell these are clam eggs or clam larvae because they're generally pink the day after. And you can see, these are microscopic. And there's just so many of them in there, hundreds of millions, that you can actually see the pink water. So then for the first 2 weeks, they stay in these 100-gallon conical tanks pictured here. So these are filled to the top with filtered seawater. We add the algae that we talked about growing last time to the tanks for them to feed on. And again, another nice picture of a veliger clam or oyster larvae swimming in the water column. They do what all babies do. They're just going to swim, eat, poop. So our job when they're at that stage is to just attend to them. So on Monday, Wednesday, and Friday, we come into the room here. We drain all these tanks down. We size grade the shellfish. We separate them out per size and then restock them according to size. I see a question in the chat. Let's check this out. What are the larvae predators in the wild? Good question. So larvae predators are going to range when they're in the planktonic stage. They're going to be anything that's swimming in the water column, everything from zooplankton, which is basically, just imagine baby shrimp or baby crabs. They're swimming in the water column. Those need to eat plankton. So they're going to eat the larvae. The bunker fish that I mentioned, which are herbivores that swim around with their mouths open, just like baleen whales do. And then once they set to the bottom, it's going to be all the stuff hanging out on the bottom, whether it's crabs, shrimp that feed on the bottom, other nursery fish that are feeding on the bottom. So they really have a lot. They're really up against a lot out in the wild.
Farley, I have a question.
Yep.
When you say that you drain these barrels, how do you, where do the new seedlings go? Where do you drain? You drain this water out into?
Yeah, let me show. There's a picture of that here, right here. So the bottom, like I said, these are called conical tanks because they're cone shaped on the bottom.
Mm-hmm.
So we pull the plug. You can see on the left-hand side, there's a hose here, which is an air line connected to a cork. So there's air being pulsed into the bottom of the tank, keeping the water in circulation, keeping the oxygen levels up. So when we go to drain them, we pull that cork and we drain them through what's called a catch screen. So this is a piece of PVC pipe with a fine mesh on it. So the water runs through and the larvae are all retained.
Mm-hmm.
This is what it looks like after the drain down. So obviously all the water is removed. All the larvae is retained on the catch screen. And then that larvae is then dropped through a tower of these sieves, large opening to small opening.
Mm-hmm.
So we pour those through the tower of sieves and we separate them out by size. So each sieve size goes into its own blue bucket. We also have an idea of how many larvae we have because we do a count in the morning. And the way we do that is we take a subsample from each conical. Say we take a half millimeter sample, several half millimeter samples per conical, count those samples and then extrapolate them out. And then we have an idea of how many millions of larvae we have to work with. And then once we size grade them through these mesh sieves, we have an idea of the breakdown by percentage. And then we restock them into the tanks Here's a picture of the larvae that are basically divided up into a certain number of buckets, and each bucket goes back into a conical tank.
Unbelievable.
That makes sense?
Yes, thank you. That's really fascinating. Was that Adam Jonas?
That was Adam.
Okay.
Yep. I think I probably just went through all this. So that during the hatchery culture stage, we do our daily counts. We feed them the algae that we grow, which is microscopic single-celled aquatic plants. Everything starts in the 100-gallon conical tanks, followed by our setting tanks, which I'll describe next. Monday, Wednesday, Friday, drain down, water change, size grade, and restock. About 2 weeks setting occurs, and I'll go over this. And then this tank here is another area of our hatchery that we call out back, which is a downwelling system. You can see the direction of the water moving through these pipes is in a downward motion. So on the bottom of these pipes is another mesh screen with a whole bunch of oysters on the bottom. And this is the stage. This is basically once the oysters set, and we'll talk about this in a bit, once they set and start to grow and become miniature oysters, we can then move them out of the hatchery into this other area in our hatchery, which is a little cooler, and get them ready for a move to the nursery. So now we're going to talk about how oyster setting works, and hopefully this will all come to make a total sense. So when oysters set, like I said before, they need to cement to a substrate. So what happens in the wild is oyster reefs, where you have oysters setting on oysters year after year, millennia after millennia, and you end up with these huge underwater mounds of oyster reef. So that's how oyster reefs are developed. You have a bunch of parent oysters, they spawn, and luckily after the 2-week larval cycle, after those larvae are swimming or cruising around the harbor for 2 weeks, they might settle right back on that oyster reef, cement themselves, and start to grow. So in the hatchery, the way we do it is we set the oysters on this powdery substance in my hand here called microculture, which is basically ground up shell. Because oysters actually, through evolution, smart evolution, oysters prefer to set on other oysters. So we take other shell material, calcium carbonate material, grind that up into small pieces, and put that on this fine mesh screen. So when the oysters are ready to set, we know when they're ready to set because of a certain size that they are, which is about 1/4 millimeter. Then we transition them from these conical tanks into these oyster setting tanks. Okay, so if you can imagine larvae like this, that's about 1/4 millimeter. It's going to be put into these setting downwellers with this culture lining the bottom. Those oysters are going to set to the bottom. Each one of them is going to find their own little piece of microculture, cement to it, and then become its own single oyster that we're all used to growing in our gardens and we're all used to seeing on the raw bar. An alternative to that is to actually set the oysters on shell bags. And shell bags are basically, if you can imagine a mesh bag full of whole shells, we can set the oysters in tanks with these shell bags in them. And then you have whole big pieces of shell with many individual oysters set on that shell. And that's what we use to grow oyster reefs or shoreline stabilization, oyster reefs, oyster reef restoration. But for single oysters, or what we call cultureless oysters, this is the way it's done. Okay, does that make sense? Do we have any questions so far?
Is there any difference in how the oysters develop if they're set on the ground or if they're in the bags like we'll be growing them?
Good question. Yeah, well, if they're set on the ground, so the reason we grow them in bags is to keep them safe, keep them away from predators. So that's one of the main differences is predation. We float them at the top of the water column because the water is warmer up at in the top of the water column, and there's more food at the top of the water column because that food, being single-celled aquatic plants, photosynthesizing plants, there's more of them at the surface of the water where there's more sun. So if you were to take a bunch of oyster seed and just put them directly onto the bottom, if they're small, they're going to get devoured. They really, unless they're a certain size, and that size that we understand is about 40 millimeters or about 1.5 inches. If they're much smaller than that, they're just popcorn for crabs and other bottom feeding fish. So if they do survive, they generally tend to grow just as well, if not a little better actually, because if they're sitting in the bottom, they tend to have a thicker shell, possibly because they're constantly opening and closing and being chipped away at by crabs. And so the ones that you grow in your garden at the surface are generally going to grow a little faster. So they're going to be a little bit thinner shelled. And there's some techniques that you can use to make them develop a thicker shell, which we can talk about later. And some of the veterans will talk to you about that too. Any other queries? Okay. This next picture is a teaser. Is this a video? Yeah, this is a video. These are scallops coming soon to a shellfish nursery near you. Those are little baby scallops, these are actually probably 1 or 2 millimeters in size. Very active. Have their foot sticking out. So we'll have those at our nursery June or July. Hopefully you guys can come by and see those. And I think that's pretty much it for hatchery nursery culture.
Farley, will they be at the Montauk Hatchery, right? Not the smaller one at Gann Road.
Which?
The scallops.
These scallops were actually, this scallop video was taken over at the nursery.
Okay.
We'll hatch them here out in Montauk, but then once they set, once they've dropped to the bottom and they grow to about 1 millimeter, and then we bring them over to the nursery, grow them over there on ambient water. And we'll talk about the nursery. I don't know if I actually do a talk on the nursery. I probably should. Well, the next talk is going to be first Thursday in May on shellfish disease, pests, predators, stuff that's really important to you gardeners, stuff that you need to keep an eye on to keep your oysters alive and healthy.
When will we be given the instructions for the new crates or the new containers?
As soon as I get them in.
Oh, okay.
Probably May, definitely not this month. Hopefully we'll have, your first maintenance weekend is May 6th. I hope to have them by then. If not, it should be beginning of June. I'm having problems tracking all that material down, but I'll let you know as soon as that happens. So what Susan's referring to is, in the past, you guys used floating cages, and we're going to switch to individual floating bags. So there'll be more on that later. So do we have any other questions about, and you guys are welcome to come visit the actual hatchery, with this background. This background helps a lot. And you're welcome to come visit the actual hatchery and look at some larvae under the microscope, check out the algae production in real life. And all this stuff should be in perspective.
What are the hours that the hatchery is open?
Good question. We're generally here definitely call beforehand, especially this time of year, because we've got stuff at the nursery, so we're gonna be running all about. But generally, figure 9 to 3 is a good time to swing
Mhm.
by.
Okay.
Definitely either email me or give me a call first. And happy to show you around, pull some samples. Really exciting stuff to look at. Larvae are always cool to look at, swimming under the microscope. Once clams set to the bottom, they're really cool to look at. Let me see if I have some other videos I can show. What happened to that? I'm looking for some other videos that I might be able to show you, but. Any other questions, folks? Let me show you. Can I drag this picture to the other screen? Everybody see that?
Yep.
Yes.
That's what I mentioned, setting oysters on whole pieces of shell. This is a surf clam shell with a bunch of little baby oysters on it.
Wow.
That's the opposite of this. Whoop, that didn't work. That's the opposite of a single oyster, which you guys all know. So here's a clam video. That's not going to drag over. Let me share this clam video. So these are a bunch of little set clams. You can see that, right?
We lost your voice. Ollie, we lost you.
Oh. Am I back?
You're back.
All right. Sorry about that. I must have overwhelmed my archaic computer with all those videos. All right. Does anybody have any other questions, concerns, comments?
What's the difference between the hatchery and the nursery, and which one is where?
Ah, good question. So the hatchery is it's out in Montauk. And that's where everything starts. So we start here in really December, starting to grow the algae, to start conditioning the shellfish. We do all the conditioning, all the spawning, all the larvae culture here. And then following that is the nursery stage, which began actually today. We brought the first batch of oysters over to the nursery, which is early because the water's still kind of cold, but they literally eat us out of house and home. We can't grow enough food to keep them here. So we cool them down. The water temperature is hovering right around 50 right now. So we cool them down, we bring them over there. They'll slow down feeding or go dormant. And then once things warm up, which seems to be happening quickly, today was pretty warm, they'll start to grow. And then as they grow, we'll transition them to the field site, which is in Napeague Harbor. And we'll grow them up so this year's oysters will grow to about 2 inches in size. And once they reach that size, we'll seed them. We seed them all throughout town from Northwest Creek all the way out to Lake Montauk. So the place to visit right now is really the hatchery out in Montauk on Fort Pond Bay. Any other questions? All right, I guess I'll let y'all go. About cocktail hour, I reckon. So hopefully we'll see y'all May 4th. Should be back in town hall that day, and we'll try another hybrid. And I'll meet with IT and suss out all the potential technical problems. Should have some stuff hands-on to show you, but it's always good to meet in person. This Zoom stuff's getting old. Yeah, any other questions? Feel free to reach out. Come for a visit at the hatchery. And we'll see you next time. Thank y'all.
Thank you, Paulie.
Thank you.
Have a wonderful holiday, everybody.
Oh, you too.
Thank you.
Thanks. Good night.
Bye.